And I think Dr. Bedford is going to join me for the presentation of the award.
Hello and good afternoon. Just a word or two about this award. The award is in commemoration of Cambridge University professor Felix Franks, who died 8 years ago. He was a highly distinguished former chairman of the Biotechnology Group, and the award is the result of the generosity of his family, who have agreed to provide the medals. The aim of this RSC Biotechnology Group Award is to encourage and recognise excellence in early career scientists in the application of chemical sciences to the study of any aspect of biotechnology. The award is made for the most meritorious contributions to chemical biotechnology on the basis of published papers and other documentary evidence over the preceding 5 years. We encourage proposals both from industry and academia and accept supporting evidence from more than one person if the candidate has not been able to publish confidential work. The award is open to all early career scientists, whether or not members of the Royal Society of Chemistry, and the definition of an early career scientist is normally someone who has commenced their PhD less than 12 years before the closing date. So the Biotechnology Group Committee are very pleased to announce that the recipient of the 2024 Felix Franks Biotechnology Medal is Dr. Matthew Jenner of the Department of Chemistry, University of Warwick. Matt receives the award for his internationally leading research into the discovery and biosynthesis of natural products through application of innovative mass spectrometry-based approaches. So I'd like to now invite Matt to give his lecture, but before doing so, I will present him with a medal. His lecture is entitled Unraveling the Biosynthetic Logic of Fungal Siderophore Biosynthesis. So without further ado, perhaps you'd step forward, Mark, and when get this lid open. There you go. So the award is a handsome gold medal, which I think we're going to try and get a picture of it. So I'd like to have it going. No, hold on. Just going to take a picture. Okay. Okay. I'll come to you, Matt. All right. Thank you.
So I'd just like to say that I'm really pleased that this is part of this session, because it's a session that's mentioned so many people who have passed on, that it's nice to see that there are younger people coming along. So
okay, well, thank you, Colin, for that really kind introduction. Thank you to the RSC Biotechnology Committee for the award. It's been a real honour to receive that this year. Thank you to the conference organisers for sort of morphing this into an award lecture. And today I wanted to tell you about two short stories related to siderophore biosynthesis in fungi. And in both cases, as Colin mentioned, intact protein mass spectrometry played a really key role in unpicking what was going on here. So I hope you can all appreciate that iron, depicted here in orange, is a really essential element for life. It's central to most cellular processes. And whilst we can source iron from our diet, most microorganisms have to source that iron from the environment. And one of the key strategies that they employ here is to biosynthesize low molecular weight iron chelating compounds called siderophores, which are secreted into the environment to capture ferric iron and then bring it back to the cell through these transporters. And that's sort of depicted here. It's this one, isn't it? So in fungi, the majority of siderophore compounds belong to the hydroxymate class, with the hydroxymate group looking like this. And each siderophore has 3 hydroxymate groups to form a hexadentate ligand around an iron center, allowing it to chelate iron. And this hydroxymate class can be further broken down into 2 families. There's the cyclic hexapeptides, which is exemplified by the ferrochrome family of siderophores, and we've got the hydroxymate groups here as pendant groups on hydroxyacetyl ornithine residues. The second family, the depsipeptides, exemplified by fusarenine and coprogen. The hydroxymate groups are sort of nested within individual monomer units to form the siderophore. So today I want to tell you about the interesting biosynthetic logic that fungi apply to assemble ferrochrome. Oh dear. Ferrochrome and fusarenine. It was meant to light up anyway. So under iron depleting conditions in the laboratory, this Aspergillus nigilans strain produces exclusively this molecule here called ferrochrosine. And that is assembled from 3 hydroxyacetyl ornithine residues, which have their hydroxymate groups, and a glycine-serine-glycine tripeptide backbone. But it's also worth noting this related siderophore, which is not produced by the Aspergillus nigilans, but other fungal strains. And this is called ferrochrome, and the sole structural difference there is replacement of the serine with a glycine residue, giving us a triglycine backbone. Now, the biosynthesis of ferrochrosine by this Aspergillus nigilans strain has been known for a long time to be directed by this NRPS here called SID-C. But unifying the biosynthesis with the NRPS gene has been quite tricky for 3 key reasons. Firstly, unlike bacteria, it's very difficult to predict the substrate specificity of fungal A domains from the sequence data alone. So we don't know which adenylation domain activates which amino acid in the pathway. Secondly, we appear to have adenylation domains missing in modules 4 and 5. But most importantly, there are only 5 condensation domains, yet 6 amide bonds are required to assemble the cyclic hexapeptide. So there's clearly some strange things going on. So in an effort to start tackling this, we first individually cloned and expressed each of the individual A domains as recombinant proteins and screened them using standard adenylation domain specificity assays. And this showed that the A1 domain activates serine, A2 glycine, and A3 hydroxyacetyl ornithine. So this allowed us to annotate the NRPS as shown here, assuming each A domain loads its own modular carrier protein. So knowing the substrates of this NRPS, we then turned to integrating this NRPS more thoroughly in vitro. And by overexpression and purification from yeast, we were able to access the entire SID-C NRPS, and we could therefore assay it in vitro. And that allowed us to control the substrates which we were supplying to the enzyme. So in the first instance, if we supply it with serine, glycine, and hydroxyacetyl ornithine, perhaps unsurprisingly, we produce ferrochrosine. If we remove serine from that cocktail of substrates, we shift production of the siderophore completely over to ferrochrome, which is what I showed you earlier, with that triglycine backbone. Omission of glycine in either of these scenarios completely abolished siderophore production. So with this relatively limited information in hand, we made 3 key biosynthetic proposals for SID-C. The first was that the A3 domain loads T4 and T5 outside of its modular context, and that accounts for the 3 hydroxyacetyl ornithine residues in the final product. Secondly, somehow module 2 acts in a non-canonical fashion to load an additional glycine residue onto the free amine of the first condensation product. And that would give us the glycine-serine-glycine backbone for ferrochrosine. That same activity could also be iterative in the absence of serine to give us our ferrochrome glycine backbone. Now, I hope you can appreciate there's some fairly big biosynthetic claims which need some experimental evidence. So we started off by looking at this intermodule loading by the A3 domain. So with a soluble form of module 3 in hand, so we've got C3, A3, T3, we figured that intact protein mass spectrometry, which we've heard a lot about in this session, would be a very good way to track where these hydroxyacetyl ornithine residues are going. And indeed, when we load this module, convert it to its hollow form, and then incubate it with its substrate and cofactors, we get complete conversion to the hydroxyacetyl ornithine loaded form. We could extend this construct out a bit further to the C4, to include the C4 and the T4 domain. And when we do that, and we run exactly the same assay, we see 2 mass shifts corresponding to 2 hydroxyacetyl ornithines loaded onto perhaps the T3 and T4 domains, but they could also be the condensed product. They would be isobaric. But the killer experiment here was to inactivate the T3 domain such that it could not be loaded with its phosphopanthine arm. And then when we do this, do the same reaction, we see only 1 mass shift corresponding to 1 hydroxyacetyl ornithine, which must have been loaded onto the T4 domain. We can reproduce these loading events using a dissected approach, here supplying the C4, T4 as a separate didomain. We still see transfer of the hydroxyacetyl ornithine. And this made it easier to extend our analysis out to the C5, T5 domain, where we also see loading. So the A3 domain can transfer or load T4 and T5, which is outside of its modular context. But a really interesting observation came when we deleted or removed the N-terminal C domains from each of these constructs, which appeared to completely abolish transfer of the hydroxyacetyl ornithine residues. And this initially puzzled us until we started to consider how the A3 domain is able to communicate with T4 and T5. And we started to envisage that the polypeptide chain is perhaps wrapping around on the A3 domain, even with contacts forming with the C4 and C5 domains, which would bring the A3 domain in proximity with T4 and T5. Now, this isn't structural biology, but it is AlphaFold. And if you do an AlphaFold model of the A3 to T5 region, it did seem to agree with our hypothesis. The C4 and C5 domains formed a very nice interface with the A3 domain, forming a platform perhaps for the T domains to access the active site. So having established that A3 domain loads 3 carrier proteins with the hydroxyacetyl ornithine, we turned our attention to this proposed non-canonical activity of module 2. And here we were suggesting that this module 2 is somehow able to load an additional glycine residue onto the free amine of the first condensation product, and that this activity is also somehow iterative. So we were able to clone and express a soluble form of module 2, and using the same approach, this time incubating it with ATP and glycine, I fully expected to see, in this case, just 1 single glycine unit loaded onto the T2 domain. In fact, what happened was we started to accumulate chains of tri- and pentaglycine appended to the T2 domain. We were able to pin this activity down to the adenylation domain, because when we inactivated the C2 domain, we got exactly the same spectrum. So that ruled out the possibility that separate molecules of module 2 were condensing glycine units together. This was simply the standalone module, the A2 domain, was somehow able to form amide bonds. Now, this isn't completely unknown. We saw that in a previous lecture, but we've never seen an amide bond forming adenylation domain, especially not being able to do this iterative activity embedded within a megasynthase. So, but we did still have one problem. This A3 domain, at least in isolation, in vitro, was forming chains of 3, triglycine chain, which was good for our natural product, but it was also making longer chains going up to 5, which would make a bigger natural product that we don't see. And we postulated here that the C3 domain, the downstream C3 domain, was gatekeeping or specifically condensing across just the triglycine product. So when we co-incubated module 2 with module 3 and all the cofactors, we started to see emergence of a condensed triglycine product with the hydroxyacetyl ornithine growing over time. We could do one extra experiment to show something here. We pre-incubated module 2 on its own with glycine, allowing it to make a mixture of tri and pentaglycine chains, and then exposed it to module 3. And in this case, it only selected and condensed across the triglycine product. So the C3 domain is very selective for 3 residues at this position. So with this new information, we're able to propose a new biosynthetic trajectory for SID-C. In the first case, the serine and the glycine are loaded and condensed. Our A2 domain then loads the additional glycine residue onto the free amine. Loading of the hydroxyacetyl ornithine allows condensation across this region here, gate kept by the C3 domain. The A3 domain then loads the remaining hydroxyacetyl ornithines, which allows it to zip it up into its ferrochrome natural product. So I promised 2 stories. So that closes the biosynthesis of ferrochrosine here. But in the last few moments, I just want to talk to you about fusarenine C, which is an exemplative member of the depsipeptide class. And fusarenine is assembled from 3 cis anhydromevalonyl hydroxyornithine units. We've got this hydroxamate unit here nested in 3 of those monomeric units. And this slightly different Aspergillus fumigata strain is known to exclusively produce this siderophore. The NRPS, responsible for its biosynthesis, again looks slightly non-canonical, must have some nonlinear behavior. But our proposal, based on observations from SID-C, would be that the A1 domain is able to load both T1 and T2 with cis AMHO units. This could then be condensed, each noting that we're forming ester bonds here instead of amides. This is slightly unusual activity for a C domain. So we get our first condensation product, another loading event, another condensation, and then the CT domain presumably was gatekeeping this process to ensure that only the trimeric species is cyclized to give the siderophore. If you want to know how these monomer units are biosynthesized, I suggest you go and see Joe's poster, P48. So this needed proving, and again, intact protein mass spectrometry came in very useful. We used the same trick as we did for SID-C. We inactivated the first T domain, so we couldn't load a pantothene arm on, and the A1 domain was clearly able to load 1 AMHO unit onto T2. So that was stretching across there and loading that carrier protein. Inactivation of the C2 domain gave us a mixture of singly and doubly loaded AMHO. And in vitro biochemistry on the purified NRPS validated our mass spectrometry-based findings. Mutation of the T domains and or the C domain completely abolished siderophore production. When we deleted the the C terminal releasing condensation domain, it abolished production of fusarenine C, but it did start to produce linear hydrolytic versions of the trimer and the tetramer, which could be rescued by putting the releasing domain back in trans. This this truncated form of SID-D, which lacks its releasing domain, is really useful for intact protein mass spectrometry and probably gave us the most satisfying mass spectrum of all of this work. Here we could see emergence of a mass corresponding to a singly loaded T1 domain and the trimer species coming into existence on the T2, but it was also binding iron, which was quite difficult to omit from the reaction. So this represented a fully assembled but fully stalled NRPS, which we were capturing in the mass spectrometer. So with that, I want to try and unify some rules that we've learned from fungal NRPSs. Firstly, adenylation domains can do intermodular loading, so they can load carrier proteins outside of their modular context. Perhaps this makes them more atom-efficient enzymes. You don't have to make adenylation domains if you don't need them. Secondly, adenylation domains can form amide bonds, and that can be an iterative process, and they can be found embedded within NRPSs. And thirdly, because of these crazy adenylation domains, you need some condensation domains to do some gatekeeping activity to ensure that biosynthetic fidelity that we see. So with that, I'd like to thank some people. A really huge thank you to Yang Hai, who not only set up these projects, but he did a lot of the underlying biochemistry, which I've shown you today. Thank you to Yi Tang, who introduced me to Yang in the first instance. He really oversaw the work, and he was a fantastic host when we visited just before the pandemic hit. Thank you to members of Wenjun Zhang's lab, Rachel Liu's lab, and Neil Garg's lab for some substrate synthesis and help with some assays. Thank you to my lab group, my fledgling lab group, mainly for putting up with me on a daily basis. And thank you again to the Biotechnology Committee for the award. It's been a real honour. So thank you. I'll take any questions.